Thank you.
Thank you. So I'd like to call on Mary Opio from Kenya Marine Fisheries Research Institute, who is part of the INNOECOFOOD project. And she will tell us a bit more about our eco-hubs in our project. So
thank you so much, Nazlin.
Yes,
Mariana,
welcome,
you
Mary.
Right? Thank you. Yeah, so my name is Dr. Mary Opio. I'm a senior research scientist at Kenya Marine and Fisheries Research Institute, based at the National Aquaculture Research Center. So I'll present about the sustainable production of fish, spirulina, insects, and also fish feeds in an eco-hub setup. Our project lead, Dr. Antonio, had already shared the schematic drawing of the eco-hub, which will be set up in 4 countries which are implementing this eco-food project. So in sustainable production of fish, we'll be using recirculating aquaculture systems. That one is RAS. At the same time, we'll be using the in-pond recirculating systems. So just a brief introduction into aquaculture. Like aquaculture is very important for food and nutrition security and employment globally. And in sub-Saharan Africa, it is one of the fastest growing food production sectors, and with the production of 753,000 metric tons, which is just 0.76. Although it is the fastest growing, but the production is still very low. And that one is what motivated us to this project, so that at least we can enhance production in a control system, which is climate smart. At the same time, it is environmentally friendly. So the recirculating aquaculture systems, they are closed loop systems that recycle water in a controlled environment. At the same time, they conserve energy. That one means that we will be relying on green energy using solar panels. At the same time, in case we don't have the green energy or in cloudy periods, then we can be using the main grid. But because it is recirculating systems, the energy will be conserved because the water will not have to be preheated again once it is heated and it has gone through the closed loop, the entire cycle. Then the in-pond recirculating systems, it is a cheaper and more efficient production system than even RAS, because we have like small raceways in cells inside a pond. And it is very efficient because you use like the entire pond to power the entire system. And the feed which is being used is very little, and it is able to recycle water and also to do water treatment in a pond. And this one is being championed by our scientists at CLA in Egypt. And in these systems, the filtration, mechanical, and biological water treatment technologies are used to maintain water quality, which minimizes the environmental impacts and it maximizes the production efficiencies while reducing water usage. At the same time, utilization of less labor, because they are automated. It means that you can only have 1 person instead of having like 5 person in a normal pond situation. And also the energy conservation. So you don't have to spend a lot of money in paying for power. Next, Mariana. So fish are highly nutritious. And if we compare production of fish with other animal production systems, it has a very low carbon footprint. That one is 2.9 compared to if you are rearing cattle, which is 30 kilograms of CO2 per kg. Then the low water consumption and even the low FCR is very impressive in fish, because in fish is whereby you can get like 1-to-1 FCR if you are rearing fish compared to like livestock or even pork. Next. So in the eco-hubs, we'll be having RAS systems in Kenya, Ghana, while the IPRS will be having them in Egypt and Tanzania. And as I said before, the IPRS will be in pond setups of 4,000 meters squared earthen ponds with cells of raceways. And in the RAS systems, we will have 10 RAS systems for growing tilapia and catfish. And the water quality control will be done by the IoT sensors, which will be fixed in these systems to help us monitor ammonia, temperature, and oxygen level. And this will also help us with the fish welfare to detect whether there is anything which is happening to the culture units. And we expect to produce around 4 tons per month from the eco-hubs. Next. Oh, that one is just a photo of an existing RAS system, which is in Kenya, a place called Kamodanga. And next. Then the feed production in the eco-hubs will be having locally available feed ingredients being used in these eco-hub, because most of them are located in rural areas, whereby we have agricultural activities going on, and we'll be able to source the ingredients from the communities that are around. At the same time, we'll be having the production of the BSF and cricket in these units. So that will mean that we'll be having ingredients which are enough for the production of feed. And the feeds are sustainable because when we use BSF and also we use spirulina, which is produced in the eco-hub for this feed production, the cost of production is not very high, and they are also climate smart because spirulina has got very low carbon footprints, the same with BSF, and their protein levels are very high, meaning that they are able to give us sustainable quality feed. And the research that we've been doing has shown us that BSF combined with spirulina can replace fish meal up to 100%. So it means that when we are using BSF and spirulina as the major source of protein in the feeds that we'll be producing in the eco-hubs, then there will be no need of using fish meal, which is not environmental friendly. Next. The spirulina production in the eco-hub, for the people who do not know spirulina, it is a photosynthetic blue-green cyanobacteria, and it has more than 60% crude protein. It is very rich in fatty acids. It adds all the minerals and even the amino acids which are required. Like a normal human being can survive with spirulina like throughout the day. If you take spirulina, then you don't eat any other thing, you shall have gotten all the nutrients that your body requires, including the omega-3, the omega-3 fats, and it is considered as a superfood. And it uses very little water to produce, like 2,100 liters per kg of protein produced from spirulina. When you compare that, if you are producing soybean, or you are producing beef, which needs a lot of water for irrigation, then spirulina uses only 25% of that, and 2% compared to beef production. And then in this project, we've been able to characterize different spirulina strains from the wild in Kenya, in Ghana, and also in Tanzania and Egypt, to be able to identify the best strain which will be isolated, and mass culture will be tried first before we mass culture them in the eco-hubs, so that every country will be producing the best strain that is required for maximum production. Next. For the insect production, in the eco-hubs, we'll be producing crickets and black soldier fly larvae. And these are insects which will be producing in a way that they are certified for human consumption and also to be used for feed. for feed for fish and also feed for other animals. And to combat food insecurity, we are moving to eating insects because crickets contain up to 67% crude protein and BSF up to 50% crude protein. When they are formulated into products which are very good, like we have cricket flour, which can be used in porridge and also in cookies, then they are very high nutritious food for children, for nursing mothers, and even for anybody else at all, across all the age groups. They can be consuming these insects. So when producing insects, it is very low rearing technologies and also using the local materials. So it means that it is cheaper to produce these insects, and they are also environmentally friendly because you'll be using very little space and will be using little water. Then chitin and chitosan, which is gotten from the legs of these insects and even the people casing for in these insects, can be used for antimicrobial usages, like they can be added to the feeds to ensure that the immunity of the fish or even the animals is higher. In the insect also, we'll be using IoT to monitor the insect's behavior, welfare, and health. And the byproduct from insect culture, like frass, can be used by the local community for as a manure for their agriculture, for their agricultural produce. And when frass is used, it means that you minimize pest attacks in these crops, and also it leads to very high yields. Next.
Well,
that one is all that I had. Thank you. I acknowledge the Horizon Europe and also UKR grant for this work. Thank you.
Thank you so much, Mary, for a very clear presentation of everything we're trying to do in the EcoHub. This will be complemented now with the next speaker, Benjamin. Well, next two speakers, Benjamin Minter and Stephen Barrion. Benjamin is going to talk about the sustainable processing of the products that you just heard about, fish, spirulina, and insects, by drying, and Stephen on extrusion of these products. So can I ask Benjamin to share your slides, please? Benjamin, are you there? Can you hear me? Mariana?
Yes, I don't see Benjamin here.
Maybe Stephen can talk about his. He's
Hello.
here. Oh, I can hear somebody.
I can, yes.
Hello.
Hello,
Yes, yes.
Is it Benjamin?
Yes, Naslin.
Oh, great. Okay, thank you.
Okay, so do I share the slide or Mariana?
Yes, please. Or Mariana, are you sharing it, or you can share your latest one, whatever you like. Are you sharing it, Mariana?
No, I thought Benjamin is trying to share, but I
Benjamin,
can
are you
share.
sharing? Lovely, thank you.
Great, thank you, Mariana. Thank you, everyone. Good morning. Okay, so I'll take the first part, focusing on the sustainable processing of fish, spirulina, and insect products. And as part of that, my colleague Stephen from UNAM will also look at the extrusion aspect. Mary already laid the foundation, and so I will build up from there. Mariana, please can we move to the next slide? So just a quick flashback on what sustainability is. I put this picture there. To simplify it, it involves three things or three key factors. We talk about money, we talk about the environment, and we talk about people. So sustainability basically focuses on ensuring a balance between we making money from our activities and protecting the environment, and also making humans happy, that there wouldn't be any adverse health effects on humans. That is it. So we move on to sustainable production in the INNOECOFOOD project. So Mariana, I think you can release for that for the next slide. Yeah, so let's move so that we get everything. I thought I was going to control it from here. So please move on. Yeah. So as Mary indicated, so we'll be looking at using the produce from the production stage, and then we will be drying this produce. So the produce we'll be focusing on is what Mary talked about. That's the fish, the spirulina, and insect. And for food, we are focusing on using crickets. And so when we get to the drying stage for fish, we will be using innovative technology that is using the multi-purpose dryer that is developed by partners in the project, which is powered by solar. And so that will be used, and that for spirulina also we use another dryer, that is the Sparky dryer, which is also solar. And so for spirulina, we'll be comparing the Sparky and that of the multi-purpose dryer to see which one works best. And for insects, the Sparky dryer will be used, which is also uses solar. And the interesting aspect of this is that we are talking about sustainability, and we want to ensure that our products are safe during that processing stage. So we'll be using AI and Internet of Things, which will provide us real-time monitoring of our produce during the drying stage. For example, temperature and humidity is very key in this drying process. And so we will be remotely be able to monitor the temperature and then the humidity in the drying system. So for example, low humidity is very relevant, very key in drying process, so that it will prevent spoilage. So we could have real-time monitoring that will indicate what is happening, and then the appropriate corrective actions could be put in place so that we have our produce that we are drying does not get spoiled, and it will come out well dried, and it will be safe for consumption. From these, where there is a need for us to cause some freezing or chilling, there is also the ice making machine that is also solar powered that will be used to complement the process. From the drying stage, we move to the very first intermediate products that we have the dried fish, which will be either whole fish or filleted fish. So we'll check which one works best. And then the filleted fish will be used for the fish powder production. That takes us to intermediate products too. And for spirulina, we have the dried spirulina, and then we will have the intermediate product two, which will be the spirulina powder. And for the crickets, we'll have the intermediate product two, which is also the cricket powder. Of course, some minor processing will go in there. And in all these, we are not leaving quality and safety out. So the quality and then the safety aspect of the product will also be checked and then used for other processing. Can we move on to the next slide? Yeah, let's move on. Yeah, so we pick intermediate product 2, which is the pulverized form, that is the dried powder form of the various produce that has been dried. And then now we look at the blending behavior of these novel ingredients in addition with other relevant ingredient that goes with specific product. And then we will now use them to fortify conventional products specific to individual and partner countries, what works best for them. So different partners from Kenya, from Ghana, are looking at different products. So products like conventional products that we will fortify with these novel ingredients include biscuits, corn sticks, cookies, porridge, we have sausage, we have soup, and then we have the NutriBars. NutriBars being developed by Mekerere. I think Agnes will be talking about that in section 3. And then we have the extruded snacks, which I indicated that my colleague Stephen from UNAM will be looking at that. And the safety and quality aspect is also being considered. And interestingly, the initial checks that is being done in this formulation shows that using these ingredients improves or enhances the protein content and the mineral content of this product. And of course, the fat is of good benefit to humans also. So the results are very pleasing, and I see that it is very good venture to pursue. So let's look at some of the product that has been developed so far in picture form. So we have the Jacout extruded insect cricket powder, and then they have the porridge also, and then we have the perch cookies, and then they have the bread rolls as well. And this is made with cricket powder. And then we have the CSR-FRI high quality cassava flour and potato flour biscuit containing spirulina and fish. That is one of the products. We have one that also contains the crickets and the combination thereof. We also have the CSR-FRI corn sticks containing.
Oh, I think we lost him. Benjamin, you there? Shall I finish it off for him? I think so we can move on. Okay. So thank you, Benjamin, if you can hear us.
Yeah, thank you, Naslin. I think
oh, you joined.
Done. Yeah.
Okay, I knew it was your last slide, so don't worry too much.
Yeah,
so we had the extruded, the corn snacks, did you mentioned, and you were coming on to soups, I guess.
Yes, and then I guess you mentioned that the sensory evaluation are very acceptable. We got very acceptable results. The sensory evaluation for all the products that are being developed. And the shelf life also shows very interesting results. The microbial activity of the novel ingredients are showing some interesting results after a few days during on shelf. Thank you very much.
Thank you so much, Benjamin. Very clear presentation. Thanks. And now we have Stephen Barrian from University of Namibia, who is going to talk about extrusion activities there and some products that they're making with insects and spirulina together with staples.
Thank you, Naslin. Should I share, or is Mariana going to share?
You can share.
Okay. Morning and good day to everyone. My name is Stephen Barrian. I'm from the Faculty of Agriculture, Engineering and Natural Sciences. the Department of Food Science and Systems at the University of Namibia. I'm going to present to you some of the work that we're engaged with under Work Package 6 or Task 6.11, solar dried and extruded food products. I'll be speaking specifically on the extruded food products. In Africa, 1 in 3 children experience food poverty, and this is due to the inequity, conflict, climate change, and up to 50% of them suffer from severe malnutrition. And this is reported by UNICEF. Now, in Namibia, even though we don't have conflict, we have a large income disparity between the haves and the have-nots. So that's inequity. And climate change, Namibia being one of the driest countries in Southern Africa, this is certainly a problem. So as in many African countries, food insecurity is a grim reality. Namibia has a low population of 3 million people compared to other countries, but already 1.3 million people have been categorized as food insecure, and this is straight from a report of the World Food Programme. Now, to address this kind of food insecurity, one needs to have a form of diversified and nutritionally balanced food, and this can be developed using extrusion technology. The application of extrusion technology is where you add or you apply it to raw material, and you can develop a wide range of nutritious food. What extrusion is it's basically an intensive operation which combines high temperature, pressure, shear force in order to manufacture products to a desirable texture. So basically, you are cooking, you are mixing, you are kneading raw material and manufacturing a product after it gets through this extrusion process. Now, there is a great interest, specifically in health foods, that have resulted from a variety of processes, and especially once they are served in a ready-to-eat form. So, what we'll be looking at is adding value to our drought-resistant crops. As I mentioned earlier, we are one of the driest countries in Southern Africa, and the type of crops that can withstand our low rainfalls that we get include these drought-resistant crops like pearl millet, sorghum, and to a certain extent, maize. So what we're engaged in is we have designed, in conjunction with a technology partner, an extrusion process that's adaptable to our current local conditions. From the schematic diagram and the 3D presentation, you can see we have a screw extruder on the left. We have a counterflow cooler and dryer on the top right. And once the raw material enters through there, the extruder gets cooked. It then gets cooled and dried, and then it can be passed. Either it is already ready to bag, like in the case of making snacks, or it gets passed through a hammer mill where we aim to make instant porridge. So I thank Benjamin for introducing the type of intermediate products and products that extrusion can achieve. In this particular case, we are looking at both making instant porridge as well as snacks. So snacks doesn't necessarily have to have such a negative connotation to it. You can actually, the aim here is to actually develop a healthy type of snack, which will complement the diets of children. At the bottom, we have a mixer, a ribbon mixer, where we aim to either mix the spirulina powder or the dried fish powder. But under this research, we have to still identify which is the exact unit operation where we'll incorporate those powders, which Benjamin indicated in his previous presentation. Then we have a flavor or coating drum. Typically, they would use artificial and synthetic type of dyes. If need be, we'll approach that, but the aim here is to use more natural type of ingredients, and in this case, spirulina and dried fish, and if possible, also the dried cricket powder. The pearl millet and maize and sorghum, which is mostly grown and utilized in Namibia, these have a low protein content. And this is the reason why we've been looking at all these alternative protein sources where we can fortify and improve the protein content of the products that we'll produce from these cereals. Currently, there is no instant porridge developed from pearl millet or maize and sorghum grains that incorporates spirulina, low-value fish, and insect powder. So Namibia has this challenge where a lot of our fishing companies, they land a lot of fish, but in the process, they have a lot of bycatch. And a lot of the fish that do not conform to the market size have a tendency of just throwing it overboard. But now the Namibian government has put pressure on them that they have to land this so-called low-value fish, and now they're trying to look at how they can add value to it so that they eliminate that specific environmental problem. So the research that we are undertaking is aimed at developing an instant porridge, a breakfast cereal, or snacks from pearl millet, maize, and sorghum, and to evaluate its nutritional composition. It's one thing to evaluate its nutritional composition, but of course, it needs to be palatable and it needs to be accepted by the population. So this is one of the areas that we'll be focusing on to produce a product that will be appealing to the large population while catering for the nutrition deficiency problem. Our counterpart at the Jomo Kenyatta University in Kenya, already as indicated in the previous presentations, they've worked on extruded products where they've added insect powder, cricket powder specifically, to the porridge. So here's a photo of some of the trials we've run, where we've looked at adding spirulina to the different cereal grains which I've presented. We have some preliminary data where, interesting enough, we could reach levels of 16% by fortifying maize meal with 5% spirulina. And if you look at a commercial brand instant porridge, a brand which I grew up with as a young kid, and this is specifically the wheat-free brand, which probably caters for those who have issues with celiac disease and they can't consume gluten type of product. They report that their type of product contains 16.1. So we have some promising results in terms of addressing the deficit of protein in these local cereals. And then, of course, we have to look at the other important macromolecules as well as the minor nutrients. But for now, this is what we've managed to achieve. Okay, so these are my references, and we'd like to acknowledge the funding that we've received from Horizon Europe, as well as from the UK RI, because the funding is what drives us to conduct specific research. I thank you.
Thank you very much, Stephen, for very interesting presentation on your using advanced extrusion technology for making high protein snacks and food for especially for malnourished kids and feeding programs in schools and so on. So thank you, and we'll hear more about some of these aspects in the other sessions as well. So you'll get a more overall picture of what's going on in the INNOECOFOOD project.
Thank you,
So
Nesli.
our last speaker in this session 1 is Adrian Shortbrook. I'm not sure if I pronounced your name correctly, but
Something like
welcome.
that.
Something like that.
Thank
Very
you,
welcome.
and hello, everybody. I share my screen.
Yes, please.
I just have to
Oh,
replace
you have
the
to remove
current
the UNAM
screen.
one. Yeah.
Yes, exactly.
Yeah, I think Mariana can do that.
I don't, okay.
Yeah.
Okay. Normally it should work. Now launch. Do you see the right screen?
Yes.
Okay,
Thank you.
perfect.
Yeah.
So hello everybody, and I'm taking back the word for Agroecology TRANSECT, which was introduced by Bertrand and followed by Stephen. And so I'll introduce a part of the project that was dedicated to having a portrait methodology, so trying to portray the agroecological initiatives that we call innovation hubs, and of course to always to support this transformative change. Well, just to give a broad picture during this presentation, I'll speak, of course, of the methodology, some results that we had, and show a bit an example of results we had for each innovation hub, and then also preliminary results between innovation hub, trying to see the transversal analysis of it. So just to start and to put a bit the context, so the portraying methodology, well, for each innovation hub, the idea is that agroecology TRANSECT is a travel companion. So they mean that they have an history before, and they, of course, will have a future. And in this timeline, well, the portraying methodology was really trying to capture what they are at the beginning of the project. So looking at their history, but also how they enter the project. And with the aim really to try to build a shared understanding of each innovation hubs that was present in the project. So for this methodology, well, it was divided in 4 different pillars, and it was mainly based on semi-directed interviews and literature review. This methodology was, so if I go into the first pillar, which is the context, was dedicated to trying to understand the specific contexts. So trying to see in terms of biological environment, knowledge, society, policy and governance, economy, and the farming system, where they are and what's happening around them, just to understand the context of each innovation hub. Then the second pillars was dedicated to the relation to the 13 principles. So here I take back the 13 principles are as they were built by the high-level panel of experts. So it's, I would say, a broad accepted definition of agroecology that we use in our methodology. And these 13 principles were taken for each innovation hub. The idea was to trying to have a self-evaluation by the innovation facilitator, so someone that is, I would say, coordinating the innovation hub, or at least be the contact point with here the project. And the aim was to have for each of these principles, trying to see in each innovation hub where they are at the currently. So it's a self-evaluation. So on 1 to 5, where they see themselves, and then try to see what are their future expected scores. So trying to see where they want to go, because of course each innovation hub have some more focus on some part of agroecology. So it was trying to capture where they are and where they want to go in terms of the certain principles. And so that was for something made reflect on themselves for each innovation hub. And now we can also trying to take from those, trying to extract some quantitative results. And we, as first preliminary analysis, which is transversal between the innovation hub, trying to see if there are some, I would say, a correlation, positive or negative correlation between the different principles. And so that's a part of it. And we can see here on the screen very quickly that there are some negative and positive correlations. So maybe it's interesting to develop that in a further project or research. A third pillar was the actor network. So thanks to interviews and the document that we gather, we try to build for each innovation hub their actor network. So as you can see on the right-hand side of the slides, it's for the Danish innovation hub of our project. We can see that each node is one stakeholders of the innovation hub. So we have, for instance, at the centers, stakeholders related to advisory world. Well, the idea is to see then which are the relationship with all the other stakeholders that are present. So a node is a stakeholders and an edge is a relationship between the different actors. And so with the aim to see which are more central at the external, etc. And again, we did it for each innovation hub, so they have it individually, but also now we are interesting and we are under development of it, trying to build also a transversal knowledge and trying to see, well, how can we compare it? And here we can see, for instance, we use a typology of the different stakeholders to see how they are different from each others. So it was one of the results we gather. And then the fourth pillar, which I would say is one of the more central of our methodology, is the pillar related to buyers and levers. And here again, we have a kind of a map of it, but we are not anymore in the actors. Each nodes here identify or a barrier, so something that blocked or that was bad for the development and the I would say development, but also day-to-day work of each innovation hub. And it was also here. It's in red, sorry. And then also the levers, so which are more positive aspect, things that help them in the development, which are in green, and edges are representing the effect that they may have on each others. And so here we try, again, you can see on the right-hand side an example of the Danish case study, Danish Innovation Hub. We try to gather them, trying to see which are the, I would say, causality between those buyers and levers, sorry, of positive or negative impacts. And so, of course, it helped them in their own development to have an understanding of it. But no, yes, here just a small example of it. What could be those buyers and levers? Well, if I zoom on one part of this scheme, for instance, one barrier is the fact that there is still contested knowledge about conservation agriculture related to carbon sequestration. So it's something that they say during the interviews, which was challenging for them, or the current legislation, which was apparently something also blocking. But we can see that there was also a positive aspect. For instance, the fact that there is a national university that was taking that into hands and trying to building about conservation agriculture and transdisciplinary research, or the fact that there is this collaboration in terms of between social scientists and the advisors. So as you see, some things that may be related to each others and that have positive or negative impacts. And for not entering into detail, but then with these maps, we could draw some metrics. So trying to see how they influence each others, which one were the most blocking, for instance, if they have a lot of negative impacts on others, etc. It was also something we use, and we can then take also some transversal results. So what were the bars and the levers that came more often from the different innovations? Well, for instance, in terms of knowledge and information, there was a lack of practical knowledge on agroecological practices. It's something that often come back from the barriers. Also something else, which was the advisory and research role that's a bit new for the in terms of agroecology. So not someone that is saying you have to do that, but someone which is co-constructing. It's also something challenging. In terms of policy, it may be more the inconsistency, which was problematic. In the economic market, sometimes they're trying to have and to look for this economic profitability in the current situation, which is and current markets, which are not always easy and fluctuable, which is a challenge. Society and culture, for instance, well, the lack of awareness that we can have in terms of more the citizens, etc., etc. I'm not going to say all of those, but there were some key challenges that came out. Well, now to have some key takeaways to take from those from this methodology. Well, first was The aim was really to have this shared understanding and to have this effective interaction between science and society. And I would say science in terms of agronomy, but also social science. As I presented, there was those 4 complementary pillars. And of course, the idea was really to have this scientifically robust, but also locally pertinent research to have and to guide this transformative efforts during all the project as Stephen presented. And now I thought that it would be interesting as we have those just recently a roadshow on barriers and levers that also Bertrand introduced and speak about a video. Well, here it is, just a small extract of it to raise the voice of some of our practitioners that are important in our project.
So an innovation hub is a place where farmers can experiment.
Do you hear the video? Just to be sure. Okay,
yes.
Perfect. I can manage it. Oh, why is it not working? No, not already. Thank you for your attention. Sorry.
So an innovation hub is a place where farmers can experiment with the new things on agroecology they want to try out, and we work in arable farming in the Netherlands. Our strip cropping farmers, which we work with, they really like this way of farming, and they see quite good results in that their harvests are stable or even better, and they also see good results in the biodiversity on their farms.
The ambition of Inspira Territorio, which is the name of our innovation hub in the Sierra de Aracena, is to support an agri-food system that is as much local and agroecological as possible.
Our innovation hub is one of 11. We aim to become a knowledge center for small sustainable farmers. On the other hand, we also want to communicate with our customers because our original core business is a veggie box delivery scheme. What we have done is actually bring farmers together, because they deliver to us, but then realizing that it is good that they exchange knowledge, and so we are facilitating this kind of knowledge exchange between farmers.
Although we can show that agroecology has potential to adapt to climate change or to preserve biodiversity, we also know that it's not that simple in real life. Working together between scientists and farmers in a co-creation process is the best way to come to what we call actionable knowledge, which are knowledge that are more likely to come into practice.
Understanding barriers and levers is important to upscale agroecology, to understand why makes things move forward or what hinder change.
The government in the Netherlands has introduced an eco-scheme where there is an extra cap payment for farmers if they do strip cropping. And we find this really beneficial because it's really a lever for farmers to try and introduce this on their farm, and this makes them experiment, and it's really a way to get into it. So we find that policy changes really help us.
So my deep motivation to become a researcher in agroecology and also an activist is the belief deep in my heart that agri-food systems are at the core of most of our social ecological challenges currently in the world. So it's also where most of the solutions to the challenges we're facing lie, and we can change that for a better future.
Perfect. So thank you for your attention. It was a small part of the video. You can find it on YouTube. And for more information, I forget to mention at the beginning that I was helping to construct this methodology, but it was mainly Pauline Cassard, which was dealing with it in our project. I'm a work package leader more from her side. And she built first a paper, which was recently published. So feel free to contact Pauline or to read the paper. Thank you for your attention.
Well, thank you so much, Adrian, for a very informative presentation on the barriers and also the levers that you're using to improve the transition to agroecology and the benefits faced by the people. So it was interesting to see the video as well. Thank you.
It's a pleasure.
Okay, so we've come to the end of this speaker session in the first session, and we're now into the roundtable discussion. I'll shorten it a little bit. It won't be half an hour because we've kind of lost time along the way somewhere. So I'm not sure how many questions there are in the chat related to any of the speakers, but before while Mariana is looking up the questions, I'd like to invite one of our INNOECOFOOD partners, Said in Turkey. He's the one who's looked at all the spirulina varieties in different parts of Africa. So I thought it would be interesting for you to know how we went about it. So Said, would you like to share your slides, please? Or Mariana can do it?
Sure. Let me
So just
share.
a few minutes,
Okay.
please. No, not too long.
So everybody see the slides right now?
No, we just see your folder. You have to
Oh,
click on your slides.
1 minute.
Mariana, do you have it? No, but then I suppose you can
Yes.
control it better.
Mariana, do you have? Okay, I will try one more. Share. And this one.
If Mariana has it, she can do it.
Share. What about right now?
Yeah, no. Oh, yes.
No?
Yes, yes.
Yes.
And
Just go into presentation mode.
is okay?
Yes, that's the one. Yeah.
Okay.
Thank you, Said.
So you're welcome. And hi, everyone. Good morning and good afternoon. So my name is Dr. Said Yuzak. I'm a faculty and assistant professor at the Murdur Mehmet Akif Ersoy University. I'm also one of the work package leader for the optimization and production of selected spirulina species in the INNOECOFOOD project. So today I would like to briefly introduce some of the pictures and some of the experience which we got through our sampling and the fieldwork in EcoHub countries, which are Kenya, Tanzania, Ghana, and Egypt. So, and this presentation will be about the collection and sampling from the EcoHub countries. So, 1 minute, I need to just make this here. Okay. Okay. So our sampling and the field work almost took 5 months in total, and we have visited 4 countries, Kenya, Tanzania, and Ghana, and Egypt, and we have visited the most important locations in those countries, particularly for the spirulina distribution, and including Lake Bogoria, Elementaita, Lake Nakuru, Lake Oloidan, Lake Simbi from Kenya, and Lake Small and Big Momella, Lake Natron, and Lake Manyara from Tanzania, and the Lake Volta from Ghana, and lastly, Lake El Hamra from Egypt. So because those field work and sampling took 5 months, because of that and some climate challenges, and also because of the all the official requirements for visiting takes a longer time than we expect. So before we go to the visit all those lakes in our EcoHub countries, of course we will be bringing the biological materials and the water samples from those lakes and farms, and we need to we have to take we have to get all the bureaucratic documentation done, and we have to also get authorization and approval and all the permissions from the ethical committees and the national and local ministries, airport customs, and local administrative offices. So this part was the most difficult part for us to get all those paperwork done before we go to sampling and field work. And it really took almost 10 months, which we never expect that take that actually a long time. So, but we finally get all those transfer material transfer agreements for both countries, and then we successfully and safely and healthily done with our sampling and field work. So here are some pictures I'm showing. As you see here, we visit also some farms where the spirulina are produced, particularly in Kenya. And as you see here, there are the ponds where the spirulina are growing, and the people are producing the spirulina over there, and they put in the market and sell it. And the spirulina, the powders are consumed by the local people. So we also visit the most important locations in the Kenya and Tanzania, including, for example, Lake Simbi and Lake Bogoria. These are the places where the spirulina are distributed all over in the world. So those lakes are very important for the spirulina distribution, and we decide to visit all of those lakes with our partner in ECOWAP countries. And as you see here, we actually see the flamingos over there, and the flamingos are one of the most important markers for us to figure out whether that lake has a spirulina mass or not. So because of their the waste actually are consumed by the mini algal, the living organisms over there. So another farm is in Kisumu, located in Kisumu in Kenya, and which is organized and led by the school teacher, which is called the Keriko Secondary School. And as you see in those pictures, the school teachers are organizing and producing and working with their own students to make the spirulina powder to sell it locally and provide the food particularly for the primary school, the students. Another the important lakes which we visit in Kenya is Lake Bogoria. Even Lake Bogoria, by the local people, it is called as a spirulina lake. So because of that, it has a very special strain, spirulina strain in this lake. So as you see here, it was very fascinating and happy moment for us to be able to see the spirulina mass, which is flowing at the top of the water on the lake shore, as you see on the right side. All of this, the greenish, the mass, is a spirulina population, and which made our fieldwork and sampling easier than we thought. Normally, it takes longer time to search and scan all the lakes and filter all the lake water and try to able to isolate and find the spirulina strain from the samples. But we were so lucky and happy that those lakes actually had already spirulina mass and physically can be seen and also easily to be found. So another lake which we visit in Kenya called Lake Simbi. As you see here, that lake also has a very intense and easily recognized the spirulina mass on the shore of the lake. So, and again, we see the flamingos over there living and providing the food for the spirulina and also other algae species growing and living there. So we also visit a few lakes, which we thought we can find the spirulina strains over there, including Lake Small and Big Momella and Lake Manyara in Tanzania. So in other eco-hub countries, which we visit the lakes, is Ghana, and particularly our partner CSIR has a water research institute and research facility in Akosombo, and they mostly work on the fish production, and they do the main research on the fish, but also they try to produce and grow the spirulina and other algae in their ponds. And we also visit the other farms, particularly for the fish and feed production, because the spirulina basically used to feed the fish. Most of the fish farms, they have also the ponds to grow the spirulina inside. So lastly, we visit the Egypt, which is our eco-hub. Centers will be constructed, and luckily the Egypt has a very well-known and a very well-known spinach strain, which has been used for many farms in Egypt. And we visit that farm and also visit El Hamra, Lake El Hamra. So we successfully and healthily get all the field work and the sampling done in our eco-hub countries. So this is all I would like to, I try to share from my side. And thank you so much for your attention and listening.
Thank you, Said, for showing us all the work you've done on collecting your specimens, and what you're going to do with it next will be in the rest of the project. So is Nabil there? And before we go to any questions, I'll just ask Nabil if he wants to share any thoughts on the IPRS.
Yes, I have some slides, some very few slides in order to just
it's
show
just
people.
please limit it to 5 minutes, no more. Yeah.
Yes, I will make it just 4 minutes if you want.
Great. Okay. Even better.
So I just concentrate about the system, the IPRS system, to show people how it works, because I think most of us knows the RAS system, how it works. So I'll share from my side.
Yeah, in case you've forgotten, but Mary Opio mentioned the RAS system and the IPRS system in her talk, and Nabil is using the IPRS system.
Yes, just to show people how it works. I hope you see my slide now.
Yes, we can.
This is the main target or main idea of the IPRS system, which is concentrate the fish inside this confinement, which is the cells that contributes about 2% of the total area of the earthen pond, and it uses the whole water in the earthen pond in order to make what we call it self-purification of the water. So we push water through these 3 cells, which is in this corner, and this makes 2 things. The first thing is feeds the fish that it has in a natural places, which is like river, and also it pushes the all nutrients from the place that fish grow in, and push it to the quiescent zone, which is quiescent zone. Here we remove this sludge and these nutrients in order to use after that as a fertilizers, which make more benefits. And also we can put some herbivorous fish in the open area here in order to make double product or double harvest of the fish. And the idea also of the of it, which is white water units, this which is pushes water from this place in order to push it in the cell like this. So we'll go directly to the this is a water unit, white water unit, which is depends on the diffusers in the underneath there, and then it pushes air in that way, and after that it pushes like this. This is a water unit, white water unit, make like this, and pushes water like this. So it keeps fish as in its natural place, which is increase their growth, and also removes all of the nutrients, so solid nutrients or solvent nutrients, in order to make them in the quitting zone, as we said. I don't want to make a long speech, but I'm here for any discussion or any inquiry from any of you participants to answer, and that's it. I think it's less than 4 minutes, Nazaleen.
Thank you so much, Nabil Ibrahim from CLAR in Egypt. So they're a partner.
Yes.
So I think there was a question on chat, was it?
Yes, from Antonio, but Bertrand already replied. I don't know.
Okay, so you can look at the question and the answer in the chat if you're interested. Are there any more questions? Otherwise, we move on to the next session. Oh, Bertrand has his hand up. Yeah.
Thank you, Nazaleen. Thank you all for this great talk. It's a very nice perspective on how you produce fish and insects and different type of products across Africa. I'm just wondering, do you carry on any evaluation of the energy required to have this system working? I think you're very lucky in Africa because you have you have a lot of sun. Because, for instance, what I know in insect production in Europe is much less efficient because you have to keep the insects warm. So I guess you are in a better condition to produce insects in Africa than we are in Europe. But for instance, with the last system, I see that there is this mechanism to have a flow of water. And so do you evaluate the energy needed for that?
Oh, Nabil can probably tell you about the flow of energy in this IPRS pond for the fish.
Yes, for the IPRS, it depends on the air lifts, which is air blowers. So we can easily use the ordinary electricity or the solar power right now. And right now we didn't actually calculate it, but it's easily to use.
Yes, in the EcoHub, all the energy requirements are calculated. And, and then we also use AI and IoT to control all the activities. So you'll hear more about it in the next session on digitalization. I think, yeah. So if there are no more questions, then we'll move on to session 2.